Is that it, Ryan? Okay, good afternoon. Welcome back. It's me again. I'm still from the same place. So now talking more about this fantastic little gene LMX1B, and most of what I'm going to be talking about now is from studies that were learnt from animal experiments. So everything was done under the proper rules and regulations, and all the little animals were properly anesthetized. But if you don't like seeing things having done to animals, they were all mice. So saves you putting them in traps if we do experiments on them. Okay, so the NPS gene, or LMX1B, was identified as such in 1998 by a series of 3 papers that came out that summer. As we mentioned earlier, LMX1B is a transcription factor. So that's a protein that controls the expression of other genes. So I'm going to draw DNA as just 2 straight lines for just now. And we have some region of this DNA that is a gene, and the expression of genes has to be properly controlled during development so that different cell types develop in a specific way, so that your liver cells will function as liver cells, your kidney cells function as kidney cells, brain cells as brain cells, and so forth. And that means they have to express certain combinations of genes in order to function properly. So in order for a gene to be switched on and make protein, a bunch of transcription factors have to bind to the DNA and allow for expression. So we have all these little proteins bind to the DNA, and they're going to allow this gene here to be expressed. So big red arrow, this is going to work now. So this is my little cartoon of LMX1B. And so if you don't have enough LMX1B, then it's going to go away. And then this complex will fall apart. And then there won't be gene expression. So in the absence of sufficient LMX1B, genes are not going to be expressed at the proper time in the proper cells during development. And we have the consequences that we discussed this morning and that you're all too familiar with. So I'm first going to talk about the expression of LMX1B itself early in development, and we'll see how this relates to the symptoms that you suffer from because of things going wrong early in development. So these are pictures of mouse embryos at various stages. So these are the number of days of development in mice. And whatever is staining up black or dark blue is where LMX1B is being expressed. So here we see it first being expressed right in the middle of the brain. And this, just from the turning of the embryo, is actually the forelimb bud. So there's going to be a mouse's arm. And this is early in development. Now, just a day later, because mice grow quite quickly, see here's the forelimb, hind limb, again this expression in the brain, and you're starting to see this expression down the spinal cord as well. We see now expression next day, we see expression around the eye, which fits with what Sam was saying about glaucoma. We've got more throughout the cranial mesenchyme, again down the cord, very intense staining in both the forelimb and the hind limb. The arrow here is actually pointing to the bud where a mammary gland is going to develop in a mouse, and they obviously have a number of them. And we can talk more about that later on if people want to. Again, as we go through development, again you see this expression in the eye, the limbs, all down the cord, and so forth, in the skull. Again, now this is in an older embryo, we can turn it around, you can clearly see how the expression is much more on what would be the back of the mouse's paw, and how it's much stronger staining now towards the paw than more proximally in the arm. And here, just in this close-up of the head, you can see this staining around the jaw, some around the ear, and again throughout the skull and around the eye. And this is, I believe that what we're seeing here, the expression here, has something to do with the trouble you suffer with your teeth. But we can't really take that study much further in mice because tooth development is a little different in rodents than in us. As any of you have had pets, pet rats or anything, you'll know that their teeth continue to grow. If they don't have the opportunity to wear them down, they always grow. And also, they only have one set of teeth. I think most of you who do have teeth problems, your baby teeth were fine. It was once your adult teeth came in that you had trouble. And so where it's difficult to work out exactly what's going on there from mouse studies. So this is a 14.5-day embryo that was cut like right down the middle and then looked at from the side. So again, we see this expression in the brain all the way down the cord. And now if you look at the limbs, so here you can see the sole of its foot, if you like, and the palm of its hand are poorly stained because all the staining's on the back. And here we can see staining in the kidney. So individual glomeruli staining up that Kevin was talking about there. And note, there is no staining in the gut, right? So all those GI problems, IBS and so forth that you've been talking about, that's not to do with a primary absence of LMX1B. It's secondary to something else. And we'll have some ideas about that later on. But you can see, like, all through here, no heart, lung, liver, gut, nothing in here is expressing LMX1B. And this is the normal developmental expression of LMX1B. I'm going to show you some stuff down the expression in the spinal cord now, because this relates to what we were talking about, the tingling and temperature sensation, and just fits in with a very specific change in patterns here. So this is looking in cross-section in the cord. These are basically embryos that just had the head cut off and then looking at the end of the cord. And so as we proceed through successive days of development, you can see how these neurons that start off in this position end up migrating more towards the top, and you end up with this very fixed expression pattern here. And so what's boxed here we're going to see in more detail on the next slide. And here we now show there's layers of neurons in this area, the layered 1 to 2 and 3 to 5 and so forth. And this is the staining. So Nissel's just staining nerve cells, and Trache is staining a particular type of nerve cell. And you can see this definition between layers 1 and 2 and 3 to 5 in the normal situation. In a mouse that doesn't have LMX1B, so another guy, Randy Johnson, was able to engineer the mouse genome so that there was no LMX1B gene. So we see the effects of not having the protein at all. And looking in this case, you see that there's no distinction in the layers of those neurons comparing here, and we don't get any of this staining in this region here. And what we're missing here then are those interneurons. They're like a bridge between two parts of the nervous system. And so these things in red and blue here are the small nerves that come from the tips of your fingers and your toes, and are the ones that are sensitive to like a pinprick or temperature change. And those nerves travel back up your arm or leg and into your spine, and then they connect via this interneuron to go across and then up through the main nervous system up to your brain. And the lack of LMX1B, or you having only half as much LMX1B as you should have, is inhibiting proper differentiation and development of these interneurons. So you're not getting a proper transmission of the signal from your periphery to your brain. So there's a little bit of how it's expressed during development. We'll now see what happens, how it's required for normal development of limbs. So here we see the expression of LMX1B. Here it's shown in this orangey red color. through successive days of differentiation. And this is again, and then just a staining to show you the different parts of the limb developing. And this is the hind limb of a mouse. So you'll see what you'll eventually get. Here's the thigh bone, and here's the shin bone, and the foot's down here. And that develops from these structures shown up top, with some muscle developing and so forth. And this is, and you can see how all of the LMX1B staining is all along what would be the dorsal part of the arm, okay, the top part. So we have, I think the next slide's going to show it, and again, more expression, again, very much on the top part. And it's actually not expressed in muscle, or where muscle's going to form. It's expressed around where muscle's going to form. And what's this slide? And then we saw this before. There's a precise alignment of tendons and ligaments here shown in detail in the digits, and how when these are not properly in place, that's why you have this swan necking in your fingers. And the fact that what we say, and what Kevin alluded to earlier, is that the LMX1B is required for the dorsal specification of limb development. So the differentiation of what is one side versus what is the other side of the limb as it develops. So the default pathway in the development of the limb in mammals is to be ventral. So that's having no nails, having no patella, and having all the structures that are on this side of the arm duplicated on the other. So, and that, if you could, when they do other experiments in mice, which I haven't included all the slides, but if in the animals where there's no LMX1B, the top part of the limb is a mirror image of the bottom part, because LMX1B is required for the dorsal specification. So you know that you can see you've got fingernails, and those creases and so forth are on the back of your hand. Those are dorsal specific structures. We'll see in a bit how the patella is also a dorsal specific structure. Yeah.
If you have absolutely no nails and no patella, does it mean anything worse than if you have some?
Not really. It's just a question of degree.
Is it? Okay.
Yeah. And in remember, you, as we've said before, you're all heterozygous for an LMX1B mutation. You have one normal copy and one non-functional copy. These mice are mostly generated to have no LMX1B at all, so you get a much more drastic effect. And so it gets away from all the shades of grey that we have to deal with in medicine, so that you can look at this is normal and this is obviously not, and you can see a much more black and white distinction. So again, now we can see again through this dorsal patterning. So here's the expression of LMX1B, and this is through a normal digit of the mouse. So here's this is the proximal phalanx, so it's this part of the finger here, and so there's a flexor tendon below and an extensor tendon above for bending your finger, or toe in this mouse case. So in the now where the animal has been deleted for LMX1B, so there's no expression of LMX1B at all, and now we don't have this differentiation between what's ventral with the flexor tendon and what's dorsal with an extensor tendon. There's a duplication of the flexor tendon on both sides, because there's no LMX1B to say this is top, not bottom. The default is to make bottom. So you need a signal to say, make this top instead. And so LMX1B is only expressed in the dorsal part, because that's what's going to have to change. Here's further back into the paw. So there's a metacarpal. And again, flexor tendon down here, extensor tendon above. The extensor tendon is there because of LMX1B expression. In the absence of LMX1B expression, you get duplication of the flexor tendon, and much, look where the midline is here, right? So this is the middle, and this is a complete mirror image, whereas normally you've got a lot more stuff down here in your flexor tendon, in the ventral aspect, sorry, than in the dorsal. So like on your, I'm not going to take my shoes off and show you, but you know your foot has there's a bit more meat on the lower half of your foot than on the top part, right? And there's much more stuff underneath the bones and so forth. So ventrally you have a lot more stuff going on normally, but when you don't specify this is top and therefore different, you get this duplication. And do I have the other pictures? No, I don't. You know that animals will sweat through the pads of their feet. So that's a ventral specific thing. It doesn't happen on the top part of their paws. Again, when you see duplication of sweat glands onto the top part, if there's no LMX1B, to say they don't belong. And of course, there's no claws if they don't develop, if they don't have LMX1B either. So this is just looking at a number of different genes, and their names and what their functions are won't concern us just now. But again, how their expression is changed by the absence of LMX1B. So here's a gene called Versacan, and you can see in the normal mouse it's expressed here and here, but without LMX1B it's not expressed in this area here. And that's in the forelimb and here in the hind limb. Frizzled, again, not expressed in this area in the absence of LMX1B. Changes here for nitrin and calbindin as well, comparing the absence of LMX1B with the normal situation, how this expression changes because there's no LMX1B. So a number of different genes, probably dozens, are now known to be under the control to some extent of LMX1B. Oh, this is the one I was meaning to show, yeah. So here we go. Here's a normal mouse paw from the back, and you can see all the hair follicles and stuff. And then here's the paw, the underside with all its little foot pads and so forth. Wrong way around. This is normal top, normal bottom, normal side view, normal curvature that animals have to their paws. And now, in the absence of LMX1B, the underside stays roughly the same. But now, on the top side, you've got these foot pads developing on the top of the paw. And now, when you look from the side, it's like straight like that as a mirror image, rather than showing the normal curvature that you would expect. And you can see here how the claws are developing, but here there's just little stubs. They're not proper claws because claws are dorsal specific structures, and you're not having that dorsal specification. So you can see why your fingernails we would regard as dorsal specific structures. It's the back of your hand, and that all makes sense. Now, as Steve said earlier, your patella is on your anterior aspect, facing forward. Why do we say that's on the back? Because when you were a little embryo, where your kneecap was going to form faced backwards. So here we are early in human development. So here's the arm, and there's the elbow pointing back, and you see as the leg starts to develop, the knee is facing backwards. So these start to rotate, and as the hips rotate, it turns round, and so the kneecap actually faces forward. So embryologically, the patella is a dorsal structure, just like your fingernails. So we're seeing a lack of dorsal specification with the absence of fingernails and the absence of kneecaps. So you can think, we're all meant to, originally, remember, we evolved from reptiles, and you see how lizards walk and crocodiles and so forth, their kneecaps are pointing up to the sky. Okay, so it's a dorsal structure. I'll go quickly through some of the kidney stuff that Kevin's already talked about. So again, we're comparing the normal situation on the left with a mutant LMX1B negative animal on the right. So here's a podocyte with its little foot processes, all sitting there looking great. And here is, in the absence of LMX1B, it's a improperly differentiated podocyte. Doesn't have proper foot processes and so forth. Also, we have aberrant cell-cell junctions, so we don't get this proper filtering, and there's the possibility that membrane damage and protein passing through the membrane and so forth. Two levels at which we know this is important is The glomerular basement membrane is composed of a collagen called type 4 collagen. And early in development, that's made up of two types, alpha 1 and alpha 2. And that is independent of LMX1B. There's no real difference between these ones. But later in kidney development, the composition of the membrane switches from alpha 1 and alpha 2 to alpha 3, 4, and 5. And alpha 3 and 4 are underexpressed in the absence of LMX1B. And this has been shown that people have done it, actually looked at the DNA and how the LMX1B binds to it, and shown that LMX1B binds to the DNA in front of these two genes and drives their expression. And here's some of the other genes that are known to be expressed in podocytes, and we can see that for podocin and CD2AP, that in the absence of LMX1B, there's much less expression of these podocyte-specific genes for the podocyte to develop properly. And both of these, Kevin, are associated with nephritic phenotypes, right? Podocin and CD?
Podocin for sure, it's one of the more common genetics. CD2AP, rare.
Rare. But these could be, although from what Kevin was saying earlier about, if you're going to get severe kidney disease, you may already have a mutation in one of these genes, and you'd be okay, except when you then don't have LMX1B, not only do you not have all of this being made properly, you're making even less of it because of a lack of LMX1B. So you get that double hit. So now a section through the eye of a mouse in development. So this is where the eye is developing here. This is the what's called the periocular mesenchyme that's around it, and all in pink is LMX1B expression, so that's where it's expressed in the developing eye. Many structures develop from this mesenchyme, importantly the trabecular meshwork that Sam was talking about. And in the absence of LMX1B, they don't develop normally in the mouse. So here's a side view of what's meant to be happening with the flow of fluid and so forth, and how this should drain out, and that how is part of the story of maintaining pressure. This is a normal mouse. Look here at the size of the gap here and this space, the distance here. And now in the absence of LMX1B, compare where the star is. So this is the lens. Compare the amount of space here versus here. So again, eye development is abnormal in the absence of LMX1B, presumably why you have a roughly 10-fold increase of glaucoma, because you have Patella syndrome. Now we'll talk about the brain. Okay, so this is actually looking at LMX1B expression in a rat brain. Firstly, we'll see in the, so this is roughly in the midbrain. This is the substantia nigra. You might have heard about this as the part of the brain that is destroyed in Parkinson's disease. So here we see LMX1B strongly expressed in this part of the brain. And this is a side view of a rat embryo. Again, this part of the brain, but up here looking from the side, and then seeing it expressed down the cord as we saw before. Again, the substantia nigra showing this is expression of LMX1B there, and this is a section from a Parkinson brain. So it's not that you're going to get Parkinson's disease because you have half as much LMX1B. It's when that part of the brain is destroyed, as it is in Parkinson's disease, there's no LMX1B expression. So subsequent experiments showed that LMX1B is required for the development of specific types of nerve cells. And one of these is called the mesencephalic dopaminergic neurons. And this is just showing the expression of a number of different genes and how it's at the top here is the LMX1B mutant, and down at the bottom is normal. So you can see how there's much less expression in the absence of LMX1B. For this is TH, tyrosine hydroxylase, and PITX3. Again, they're markers, a very specific dopamine genes or markers for dopaminergic neurons. So dopamine plays an important role in mood stabilization and concentration, and so the fact that these nerve cells have not been able to develop completely normally in the partial absence of LMX1B probably explains the link between NPS and ADHD and depression symptoms. So this is a cartoon about which I know very little, but it's the dopaminergic system in the brain. And there are the prefrontal cortex, the striatum and so forth, all are parts of the dopaminergic system. And come on here. LMX1B also required for the development of the serotonergic neurons. So dopamine and serotonin are both neurotransmitters. Those are small molecules that carry signals from one nerve to the next, and then on to the next and on to the next. Disruption of serotonin is involved in depression and possibly chronic pain. Mice that have been engineered to lack LMX1B only in serotonergic neurons, 5-HT is serotonin, exhibit a different pain response and do not get the benefit of SSRIs. So you can stress a little mouse out, and if you give it something like Prozac or Zoloft, it'll feel better. If you've taken away its LMX1B dependent serotonergic neurons and stress it out, SSRIs don't make it any better. Also, they have an abnormal temperature response as well. And again, this is just showing a series of markers that are known to be in those types of neurons and how they're absent in the absence of LMX1B versus our normal controls on the left. And this is a diagram of the serotonergic system. So it starts here and flows around this way, and also major efforts in the cerebellum as well. So this little diagram is to summarize really for you what we've learned from how the experiments in mice have substantiated what we thought we knew about Neal-Patella syndrome, and how when we saw things in patients we then looked at mice and confirmed it, or saw something in the mice and then realized that that really was part of the syndrome as well. So all of our skeletal stuff fits in both. We'd seen it in the patients, and then when the gene was missing in the mice, you saw the same thing. No one's actually seen if they actually develop iliac horns in the absence of LMX1B. No one bothered to look. Clubfoot, the limb development in the mice completely lacking LMX1B is so disrupted that whether they have a clubfoot or not is difficult to tell. Kidney disease, we can certainly see the changes in the kidney, as I showed you. Glaucoma, again, we see the changes in the eye. Because of what we learned about LMX1B controlling nerve cell differentiation in the mouse, that helped us think about ADHD. When I was getting lots of calls from people with Neal-Patella syndrome back in the late 90s, around 2000, as we were trying to work out, how are changes in LMX1B causing Neal-Patella syndrome. We were talking to people, and they would always say, "Oh yeah, my boy's got ADHD." And I'm going, "Yeah, every kid in America's got ADHD. Let's get on with this important nail and patella stuff that I'm interested in." And then one of the studies came out in early 2000 talking about the requirement for LMX1B for differentiation of dopaminergic neurons. And I didn't know any neuroscience at all, not something I'd ever studied. So I called across the street and said, "So if there was a problem with dopaminergic neurons, what sort of symptoms would people have?" And the guy says, "Oh, probably something like depression, ADHD." And I'm going, "Oh, really? That's interesting." So we worked up a bit more. Peripheral neuropathy, again, we saw the stuff with the expression in the for those distal nerves and how the interneurons were absent, and that we then explained in patients as well. So now what type of mutations, what type of changes do we see in LMX1B that are going to cause NPS?